So this is our agenda today. We are going to look at some assessment objectives, exam series of Cambridge curriculum. Then we'll jump back to our cell structure discussion that we were doing from our homework. And based on the cell structures we have drawn and what we know in IGCSE, we are going to compare what are some details that we are going to learn about cell structure and more complex things in AS level as compared to IGCSE. So in those complex details, we are going to be introduced to microscopes and how cells appear under different types of microscopes, and then just a little bit more detail about different structures in cell. So assessment objectives, what is the meaning of this? When I say Cambridge assessment objective, I basically mean that when Cambridge is going to hold examination, what skills are they going to test you on? So this refers to the skills that you need to show or demonstrate in your answers or in your practical. If you're giving paper 1 in your multiple choice paper, if you're giving paper 2 in your descriptive answers, if you're giving a practical, then your lab preparation, your lab plan, your calculations, your conclusions, all these are your skills. And the Cambridge board is going to assess you based on these skills. So you have to express them. You have to demonstrate or show those skills in your examination. So what type of assessment objectives come under these exams? Cambridge will assess you on 3 types of questions. The first category of question is purely based on your knowledge and understanding. So when you're learning a chapter, you're learning new objectives. You're gaining more biological knowledge. You are also understanding some processes. What is step 1? What is step 2? What is step 3 of this process? Is it a linearly flowing process, or is it a cycle? What is the name of different proteins, or where is this process happening? All these are knowledge and understanding. Facts and the reason behind why it is occurring. That is knowledge and understanding. So mostly what kind of questions will come under knowledge understanding assessment objective? They will ask you to explain, describe, identify something, or list all the examples of something under this category, differentiate between X and Y, calculate something and find out the answer. So all that is knowledge and understanding. Something that you learned newly, how much do you remember about it? And can you tell why it is happening, the reason? Or can you tell how it is happening? Explain the process. Next is another assessment objective category question is application, handling, application, and evaluation. So they will give you a data. That data can be in the form of graph or a pie chart, or that data can be a story. Maybe someone did some research in some village and they found out this information. From this information, you have to find out why are so many people dying of heart diseases. So you are going to use that information and pull out evidences from that information. What are you doing over here? You're evaluating, you're handling the information and breaking the information to decode certain things. So how are you going to do that? That is an example. So what kind of things will come in these questions? Symbols will be there, graphs will be there. Sometimes graphs will have lots of tower diagrams, and you have to explain why some parts of the graph look very tall, why some parts are shorter, just like your environmental science paper, the management paper. I think that is paper 4 or paper 3. I don't remember the number. But there is one paper in environmental science where you are doing a case study. Am I right? Yes, that is paper 2. So it's almost like that. All those analytical questions, context-based questions will be asked, where you have to apply your information handling skills. Yes, management in context. correct. Last, you can read the name only and you can guess. So these are all experiment and planning investigation based questions. This type of question can come in your paper 2. It can come in paper 3 definitely. And in multiple choice also, sometimes they ask practical questions like microscope related questions can be asked. Or some biochemical tests that you have done in lab. There are particular results and those results have a meaning. So they're going to ask that in multiple choice also. So analytical, planning, investigation related questions. These questions will have lot of words like suggest. What do you suggest? Give reason. Find out the variables. Calculate based on the data given or based on your observation table, calculate something. So these are all investigation and experiment based questions. I just added this slide so that you can get to know exactly in knowledge and understanding what things will be asked. They can ask facts, definitions, they can ask theories, concepts, any biological phenomena. Like one biological phenomenon would be bioluminescence, that is organisms that glow when disturbed. So that is a biological phenomenon. Why it is happening? How do organisms adapt themselves to do this kind of, to glow in the dark and all these things. Then other questions that can come in knowledge understanding is asking the meaning of certain terms, vocabulary, terminologies. Okay, asking why there are certain symbols used for some processes, or why do you give a negative sign behind something, or why do you always give the value in positive for certain things, those kind of questions. What scientific instrument are you using to measure certain things? What apparatus will you use in the lab to do a certain investigation? These are all knowledge questions. You should know them, then only you can do those activities. At the bottom of this table, you can see some command words written. Questions testing skills in the table above will usually begin with one of the following words: define, state, name, describe, explain, or outline. Go ahead and share with me in the chat, what do you understand by command words? You must have come across command words in IGCSE. So go ahead, do a little bit of recap. What is command word? What do you understand by these command words? You can share in the chat, you can unmute and speak, whatever you're comfortable with. Any thoughts? Yes, yes. Words that tell us how to answer a question, words that guide us and help us to understand what exactly to answer and how. I would agree that they give us hint, but they don't guide us. They confuse us also sometimes. Words and phrases used in exams and other assessment tasks that tell students how they should answer a question precisely. So command, they are commanding you to describe. They will command you to explain something, or they will command you to just give an outline of a process. That means if you are asked to give an outline, you just need to talk about the main events in the process. If you start describing every event, you may not get marks. So command words actually help you to narrow down that I know a lot about this topic, but I only need to write this much, whatever is asked. Okay, so command word will set the expectation of your answer. Yes, Muskan, I agree. With the help of command word, you can get an idea how much detail to add in the answer and how much not to. Yes. Well, sometimes stating also may not get done in 1 or 2 sentences, but most of the time stating is a very direct answer. Coming to the second assessment objective, this is handling information and solving problems. So what kind of things will be asked in under this category of questions? You will be asked to locate something, select or organize. Things will be given in jumbled up format. You have to organize them in a sequence, or a diagram will be given and you will be asked to point out and show where the process is happening. Sometimes information will be given in graph form and you will be asked to write it in words, what you understand from the graph, trends or patterns. Sometimes they will give you the data and they'll ask you to calculate mean or do some statistical calculation with the data and find out a result. That is manipulation of data. Use information to identify patterns, report any trend that you're observing, and finally give conclusion. They will also ask you to present certain reasons and explanation. So a graph with some increasing or decreasing trend will be shown, and they will ask you, why is this happening? Give your biological explanation. Why is such a trend being seen? So then you have to use your knowledge and understanding and present this in the form of an explanation based on whatever you're seeing on the graph. Prediction. Sometimes a whole scenario is given to you and then they'll ask you what will happen next. You have to predict, but you have to make a sensible prediction, not just a random guess. Because when you predict, you also have to give reason why you are predicting that this will happen. Sometimes, not sometimes, many of times, what you will learn in the book is not exactly what will be asked in exam. So maybe I am teaching you about animal cell, but in the exam, they will give you a picture of a cell which you have never seen. You have never seen that cell in the book. You have never seen it in my slides or in our classroom interaction. However, this unknown cell, when you're looking at the picture in the question paper, you can notice that there are things inside this cell which you know, which you have studied from the topic. So then what do? You apply your knowledge and try to answer question in an unfamiliar situation. So an unfamiliar situation will be given to you, but there are some things in that unfamiliar situation which related to which you have some knowledge from your biology syllabus. So you will use that knowledge to solve that problem. Okay. What command words will come under questions from handling information? Discuss, predict, suggest, calculate, or determine. And lastly, assessment objective 3. These are experimental skill and investigation questions. Here, you will be given space to plan an experiment, write all the steps, or you will be asked to do a chemical reaction or a lab and record the result. Record in such a manner that you will be able to present it to someone, and whoever sees the table will be able to understand what you are trying to say. Or you have to do measurements. So in biology, the measurements mostly means you have to measure some size of different types of cells under the microscope. You have to give estimated or approximate size or exact size. If you're doing some field work, that is calculating different types of species may be found in the Apsara ecosystem, then you might have to measure the height of the plants or insect, whatever you are investigating. So how are you doing that? How accurate is your method? That will be tested. The quality of your data. How are you making sure that your data is reliable? All that will be asked. Are you being able to evaluate? Means, are you being able to check whether your method and strategy is foolproof, or is there any scope of improvement? Are you being able to identify your own mistakes? That is also a very encouraged skill in Cambridge. It is okay that you make mistake in your investigation, but it is very dangerous if you don't know that you are making mistake. So Cambridge always encourages the learners that you make the mistake, but you also notice that you are making mistake. You should be able to be aware that, okay, I used this strategy, but in this strategy also something can go wrong. So how can I improve my strategy? So those things, those type of questions are going to come. They may ask you that suggest some steps in your experiment which could have been better, or what problems did you face and why did you face those problems. So they try to navigate the learner and make them think about where they can improve their planning. This is about third assessment. The next thing that I want to talk about is, when do these exams occur? Before I start talking about the duration and exam series, do you have any question regarding the three types of assessments? Knowledge understanding, data handling, problem solving, and investigation and experimental skill.
No.
No questions? I think you also must have come across these discussions in your chemistry and physics class, isn't it?
Yes.
Okay. Then you must have also discussed at what time in the year do Cambridge exams occur. It's very similar to your IGCSE pattern. Yes. So when are they? You can sit for the exam thrice. There are 3 opportunities. Specifically for candidates from India, they have this Feb-March opportunity. Second is May, June, and third is October, November. So these are the 3 times in the year when Cambridge exams are held. So obviously the question papers will not be same. For 3 different occasions, 3 different series of question papers are published. Due to our school's academic calendar structure and Indian college admission patterns, Avsara registers candidates for the Feb-March series. We always choose, except the pandemic year, that is last year, every other year we choose the Feb-March series, because finally grade 13 students who graduate from Avsara, if they choose to join Indian colleges, then the admissions of Indian colleges happen during April and May, or May, June. So by May, they get their results and they start their admission process. If they are applying for Indian colleges and they sit for May, June exam, they will miss a year, because May, June exam results come in August. And in August, almost all Indian colleges start their bachelor's program. Already 3 months will be over. So we choose to do the Feb-March series. Okay. With this, I wanted to come back to our discussion of cells from yesterday. So the general idea, what are the main features of cell that we studied in our IGCSE level, is something on your screen. Take a moment to look at the annotations. What do you think are these things which are written in blue? What do
they do? Functions.
The functions. And the labels written in black are the name of the structures, right? Yes.
Yes.
In the second slide here, you can see a typical plant cell. That was an animal cell, typical plant cell. Could you point out what are the differences, very striking differences between plant and animal cell here? Yes, Asba, which cell has the cell wall and which does not? Same goes for chloroplast. Plant cells have cell wall and chloroplast, but cell wall and chloroplast are not there in animal cell. Also large vacuoles. Well noted, Ritika. The permanent large vacuole is a very common feature of plant cells, not animal cells. Animal cells may or may not have vacuole. If they have vacuoles also, they're very small and temporary. So these vacuoles are like just containers. They can store things. Plant cells need to store lots of sugar and water, salt, minerals. That's why they have a permanent vacuole. Animal cells make those vacuoles only to trap food. Once the food is digested, the vacuole is gone. Okay, so like I told you in the agenda today that I will be introducing you to two types of microscope. Out of these two types of microscope, one is a compound microscope and another one is an electron microscope. Okay, this compound microscope is also called a light microscope. Can you guess and share in the chat, why is this microscope called light microscope? What is the reason of this other name? Is it because it's very light and easy to carry? Or what is the reason? Laser is present? No. No laser is present. Absolutely correct, Tamanna and Ritika. These are microscope which harnesses the light in your surrounding and uses that light to be incident on your specimen. That light will reflect and fall back into your eyes through the lenses. So it's going to use the light so that you can visualize or you can see the specimen that is kept on the stage of the microscope. So because this microscope uses light to show you what the specimen looks like, it's a light microscope. Can you tell why it is called compound? Just a guess. Of course, I'm not expecting you to give 100% correct answer, but I want to know what you're thinking. Why is it called compound? Ritika is saying that because it has different lenses. Ritika, could you elaborate a little on that point? Like, what do you mean by different lenses?
So here one of its structures is a revolving nose piece,
Mm-hmm.
which I interpret as different lenses.
The revolving nose piece has something which has lenses attached. Can you see objective lens over here?
Yes.
This is one objective lens. This is one. You can also attach a third one, but that person has not attached. Third one is still empty. So there are three lenses, and because it's a revolving nose piece, you can actually rotate and change the lens. So that is one lens. Is there any other lens in this microscope? Look carefully, everyone. You can also show me where is the other lens using the annotate button. Use the zoom annotate and show me. Circle it.
Miss Amrita, where from where we can see the eyepiece one.
The eyepiece. So in the eyepiece, there is an ocular lens, and in the nose piece, there is an objective lens, isn't it? When suppose I will give you this microscope and I'll give you a slide, a glass slide, which has some microorganisms on it. And I'll ask you, okay, put this under the microscope and see what is there on the slide. Where will you place your eye? Will you place it near the objective lens or will you place it near the ocular lens?
Ocular lens.
Ocular. So your eye will be near the eyepiece. Where will you keep your slide? Tell me the name of the part of the microscope where slide will be kept.
Stage.
The stage. Okay, now look at the left side, the actual photograph, and see under the stage. Can you see a mirror?
Yes.
Yes.
Do you know what that mirror does? If I keep this microscope near the window, then light is going to fall on this mirror. Okay. Does light pass through a mirror or does light reflect from a mirror?
Reflects.
It will reflect. When it reflects over here, this circular thing over here, you see this. This is called a condenser. Condenser has a shutter. You can open the condenser and you can close it. When condenser opens, do you know what will happen? More light will pass through. So there is a hole over here. It looks like a camera shutter like this. So if you could see the condenser from bottom, it's like this. Have you seen camera shutters? When you're about to click, they open and then they close. This is closed. Okay. Then there is a tiny button that sticks out over here. You can hold it and rotate it up or down. Suppose you rotate this button over here, then it will open. When it opens, it will open like this. See what's happening now. Now a crack. This place is open. So light can actually now pass through. Here light was not able to pass through because the condenser was shut. If you open the condenser, light can enter. When light will enter, do you notice that on the stage there is a hole and some glass is fitted over there on the stage? So light, yes, light will pass through that hole and come out from this glass hole over here. And let's say that you have kept your glass slide on this stage. Okay, so light is going to pass through your glass slide, through your objective lens, into this body of the microscope, and then through it, over here you will have your ocular lens, and over here you will have your eye. Sorry for a horrible diagram, but please don't think this is a ghost's eye. This is a human eye. Okay, so this is your eye, and here you are going to see the image. This image is forming because light is being channeled through the specimen. Specimen means what? Specimen is the object that is kept on your slide. Let's say you have kept a drop of pond water. In this, there are tiny microorganisms floating. So this is your specimen. And this is your slide. Slide is transparent, so light can pass through it easily. Okay, but mirror is not transparent, so light will bounce from the surface of the mirror and enter into the condenser. Similarly, lens is transparent, so rays are going to be refracted. Did you study about reflection and refraction in physics, 10th grade?
Yes.
When light moves from one medium to another medium, a medium which is translucent or transparent, but it is denser, then it's going to bend. So because of all these bending, finally the light is going to fall in your eye, and in your eye you have a retina at the back of your eye, and over there the image is produced with the help of your brain. So what you're seeing is, after all of this process happening, your eye is creating an image of what you're seeing inside the microscope. And all of this is possible because of who? Would you be able to see all of this in a dark room? No.
No.
Because no ray of light would have fallen in your eyes. So light microscope uses light to make the specimen visible to your eyes. Does it only make the specimen visible? No. It makes the specimen look larger than its actual size, magnifies it. So makes it visible makes it magnified. Okay. Okay. So please mute yourself. So coming back to your diagrams that you had submitted, how did you know that a cell looks like that? From where did you learn that a cell looks like this? Something that you saw in the previous slide, or something that you saw here, or something that you have submitted in the homework folder. I know that you have learned that this is how a cell looks like in 10th grade. But from where did you get this diagram? In your mind or knowledge or whatever? You studied from the textbook, okay? Any other thoughts of how you got to know that cells look like this?
Maybe scientists saw under a microscope and I put it in the textbook.
Of course, yes, because they can't do guesswork. So what you have on your textbook has to be authentic, right? So if you didn't know how to use a compound microscope, or let's say human beings never invented microscopes, we would have not known how cells look like. So the whole credit towards having such advanced scientific knowledge, so much knowledge about molecules, tiny structure inside cell, this cannot be seen with your naked eye. So because of the contribution of many scientists, biologists, zoologists and botanists on how they used to mix and match different lenses and try to magnify small objects, all of that finally became instrumental towards development of the compound microscope. And under the compound microscope, scientists noticed that earlier what they thought that cell just has a cell membrane and a dark patch that is nucleus. That is not the only thing that is there in the cell. There are many other things. So here on the screen, you're looking at actual cells, photographs taken from cells under light microscope. Have you seen moss? These mosses are tiny plants which grow like carpets on surface of damp walls or rocks or buildings or even trees. So a tiny pinch of moss was taken and dipped in a drop of water and seen under the microscope, and you can see individual cells in the stem of a moss loaded with chloroplasts. You can also see the cell wall, very thick. And because of the cell wall, the shape of the moss cells are very particular, very rigid. They are not shapeless. Yeah. Mostly in rainy season, you find these mosses in damp and humid areas. The idea is under the light microscope, you can actually observe cell and different structures inside the cell with their original color because you're using light. Sometimes, for example, if you are scraping a few cheek cells from inside your cheek and you smear it on a glass slide, when you see under the microscope, you will not be able to see anything. So that's when you know that your specimen is completely colorless. So how to see it under microscope? If it is colorless, light will pass through it, you won't be able to see anything. In order to be able to visualize something, it has to have some color. Okay, so what we do is we add some stain. So here you can see cheek cells of human beings with methylene blue stain. Methylene blue stain has been added. Can you tell me what are the darker blue parts of these cheek cells and what is the light blue part of the cheek cell? Can you identify the parts?
Nucleus.
Which one? The dark blue area or the light blue area?
Darker one.
Yes. the darker dots in the center of the cell, this is the nucleus, and the grainy, tiny dotted regions which are lightly shaded with methylene blue, that is the cytoplasm. And the boundary of the cell is the cell membrane. Tell me one thing, do these cells seem to have a definite shape like the plant cells? Do they have a particular rigid
No.
shape? No, right? That's because they don't possess a cell wall. And cell membrane is very flexible, stretchy. So once you scrape it from your cheek, it becomes loose. So the edges are all crooked. So this is how cells will appear. Just two examples of how cells look under compound microscope. Now I will show you how cells look like under electron microscope. This is the second type of microscope. So compound or light microscope used light to see the specimen. What do you think electron microscope will be using to see the image? Beams of electrons. Asba, you don't need to know all the parts of the microscope now, but if you are enrolling into the syllabus, then you will have to know. Yes. So Ritika and Shravani are saying electrons and electricity will be used maybe. You both are very close. In fact, Ritika, you are absolutely correct. So electron beams, tiny radioactive elements are blasted. Nuclear fission is done and their electrons get released from their atoms and they pass through this entire nozzle-like body. And over here, there will be the specimen. But unlike compound microscope, you can see electron microscope is very complicated. It's a gadget connected to a computer software and computer CPU and monitor. So you will not be able to put your eyes and see the specimen, but whatever specimen is there, the image will be shown to you on a computer screen. You will be able to see the image. And let's see how the image looks. Can you see the image on the screen? I don't know if it makes any sense to you. Do you know what you're looking at? What is this?
Cell.
What type of cell is this?
Animal cell.
Animal cell.
Okay. Can you pull out an arrow and show me the cell membrane? The arrows are very here and there. Exactly. Okay. Closest is Muskan's answer. Can you show me, can you circle and show me where is the nucleus? Ekta, you just have drawn a line. Only that brown part, Shilpa? Do you mean only this part?
No, the whole circle.
This whole dark patch, right?
The whole circle inside the circle.
Yes. Can you see? Okay, I got it, girls. I understood. I'm erasing now. It's become a mess. So this is the nucleus. Now look at the diagram that you have drawn in your notebook. And look at this photograph. It has the same features that you have drawn in your notebook. Tell me what is different. What is different in this photograph versus the photograph that you saw for light microscope? How cells look under light microscope? What is the difference?
This photograph has lines. Maybe
Has?
the veins. Lines.
Lines. Did you say lines or did you say something else? Arya is saying we can see some textures inside. Okay. Do you mean to say, Arya, that inside the cell, we can see some areas are dark, some areas are light, some patterns are there? Okay. Shravani is saying there are some lines for showing cytoplasm. Actually, Shravani, the cytoplasm is packed with many, many things. But when we used to draw in 10th grade, we used to just draw the cytoplasm like this. Nucleus used to nicely shade fully black or something like that. And then inside we just used to draw these dots and circles. So this used to give us a very neat look to our cell, isn't it? As if there's a lot of empty space where jelly-like fluid is flowing. Isn't it?
Also, Miss Amrita, I think this photo is much more detailed.
Yes, I was looking for that keyword. An electron microscope gives you more insight, more visibility of detailed structures that are there. Even the tiniest feature or tiniest structure that is found floating in the cytoplasm will be detected under the electron microscope, but light microscope cannot detect so much detail. Light microscope will detect structures which are of a certain amount of larger size. Of course, our eye even cannot see that, but light microscope can detect bigger structures, whereas electron microscope will detect everything. I will show you something and I hope you will recognize. What do you think these are? These things which I'm circling in purple. What do you think they are? Can you see some lines inside? Just a second, girls. Sorry, yes. So were you able to recognize what these structures are? Because I saw some of you have drawn this particular organelle in your diagram. So I thought maybe you will recognize. Any idea what this is? It looks like this. I'm just making the lines more prominent for you. Any idea what this is? Yes. Shravani, Asba, Tamanna, you are right. No, this is not chloroplast, Ritika, because this is an animal cell. It doesn't have a cell wall, remember? The outermost margin is a cell membrane. So this will not have chloroplast. This will have mitochondria. You see lots of mitochondria. There's a mitochondria here. There's one here. There's one here. This area is also packed with many. And these structures which look like black lines, these are called endoplasmic reticulum. Do you know what they do in a cell? Function? What do endoplasmic reticulum do? Their function is they help in the process of making proteins. So their main function is to do protein synthesis, or they help in the process of protein synthesis. So let me introduce you to this specimen here. Who is this? This is a white blood cell. Also very rightly noted by Ritika in the chat, you had messaged that under light microscope, we can see colors, whatever dye or stain we have given to a specimen, those colors are visible. In electron microscope, you cannot see color. Everything will be black and white because we don't use light. We use electron beams. So electron beams cannot produce colored images. They will only produce dark areas and light areas. Wherever electron beam could not pass on your computer screen, that will look black. Wherever they could pass, those areas will look gray or white because they are transparent. So the beam could pass through. So this is the image of a white blood cell. Particularly the name of this white blood cell is B lymphocyte. And the function of this cell is to protect you from diseases. How does this cell protect you from diseases? Very much related to what you do when you are fighting from different infections. Also COVID. So this is the cell that actually makes antibodies. Antibodies are like bullets for a soldier. So soldier doesn't go to the enemy territory and stops the enemy, right? They shoot. So similarly, this B lymphocyte is going to make antibodies. Antibodies are nothing but special type of proteins. And only these type of proteins have the ability to tackle any infection or foreign pathogen which is trying to enter your body. So if your body, inside your bloodstream, there is a bacteria, or there is some side of a microorganism which is trying to enter your body through blood and is damaging your cells and tissues, immediately your B lymphocytes will start making antibodies and stop those microorganisms, which are called disease-causing pathogens. So they help you fight against diseases. Now, one main common theme for your whole AS A-level syllabus is whatever you study, you have to know the structure, and you also have to know how the structure relates to what? Its function. Why is it every time you see a certain structure, you have to ask yourself, why is this cell packed with so much endoplasmic reticulum, so many mitochondria? Nothing is there for just beauty and show in our biological system. Whatever is there, it has a sole purpose towards our survival. So if there is one type of cell which is having excessive endoplasmic reticulum and mitochondria, then there is a reason behind it. The reason is this cell has to make so many antibodies at a time that it needs lots of energy, and energy comes from mitochondria. Mitochondria makes the energy for the cell, and endoplasmic reticulum makes the proteins. So this cell, cytoplasm, has no space only. It's fully packed with endoplasmic reticulum and mitochondria. So what is one thing that we are learning? Every cell in our body will not look alike. If there is one cell in your body whose function is always to make something, some protein, then it will have those structures which are protein related, protein synthesis related structures will be in large quantity. And if that cell is always active, always producing some new substance, then it will need energy and it will have lots of mitochondria in it. So just a general idea from a simple structure of cell that you knew in IGCSE. In your AS level, you dive deeper into why is the structure like this. Okay, we know that every cell has a nucleus, every cell will have mitochondria, but why? How is the structure related to the function of the cell? You have to build that connection. That was the main idea of today's discussion, that every structure that you will notice in every cellular study, there is a reason behind why that structure is like that. And you have to figure out that reason and be able to make that link. Yes, Asba, plant cells also have lots of endoplasmic reticulum because plants also make proteins. Plants may not be making antibodies like us, but they make other proteins for which they need endoplasmic reticulum. So just before we end our discussion today, I know time is up. So a quick just background knowledge about I showed you a B lymphocyte, so I wanted to also tell you where these B lymphocytes are found. They are just found in your blood vessels. Just like red blood cells, you have B lymphocytes in your blood. They are white blood cells. They are not red in color. They are colorless. That's why they are called white. They are the cells of your immune system and they are found in your bloodstream. And your body makes new white blood cells every 10 days. So where is this white blood cell made? In long bones of your body, like your arm, lower arm, your spinal cord, backbone, thighs, legs, all these long bones. Inside the bone, there is bone marrow. And this bone marrow is the place where all your red blood cell, lymphocyte, all the other white blood cells, these are monocyte, eosinophil, basophil, neutrophil. These are all blood cells. All blood cells start their journey. They originate from the bone marrow. So even right now, when you're in your class, eating food, listening to me, or doing anything, your bone marrows are constantly making red blood cells. And they're making new white blood cells also. And like I told you, function of these white blood cells are to make antibodies to fight against diseases and protect your body from any ill health. So recall your diagram of cell, and this is a picture of all the details of the cells that scientists can see just because of an invention of electron microscope. So we'll close the class with a note of appreciation of how much detail in biology, in the field of knowledge, can be learned when new devices are invented. Imagine there would have been no microscope in our world. We would have not known anything that are not visible to our naked eye. And we would have assumed that all the organisms around us are they just have bigger organs. They don't have anything called cells. And with the help of electron microscope, we also got to know that inside the cell also so much activity is going on. So many organelles or structures are there, which we did not know before. And electron microscope got invented in 1940, which is not very long ago. So after that, the whole field of cell biology and microscopy just boomed because a lot of research came up, a lot of understanding became clear about so many processes, life processes. So this is just a computerized diagram from the electron microscope image. We will continue to learn about each and every structure from the next class. Okay. For today, I will be sharing a link of reading with you through the mail. Please make sure that you do that reading. It's just a one-page reading. Yes, life looks very simple from outside, but the more deeper you dive in, it gets more complex and complex. It's much more complex. So this is the idea, to just appreciate so many details which we were unaware of without the electron microscope. Okay, thank you everyone. Later, please check your mail and it will be on mail. I will share the link on mail. And similarly, after reading, you can share your answers in the Google Drive folder.
Bye, Miss Amrita. Thank you.
Bye-bye. See you tomorrow.